So today's subject is variant analysis, and by variant analysis, we mean what happens within a sample, within an individual. We are not talking about differences between different genome assemblies of SARS-CoV-2. We're talking about next generation sequencing data from a single individual and trying to assess how much variation we have within that individual. It's interesting for a number of reasons. Well, first of all, it allows you to validate assemblies, and we know that some of the GZ8 assemblies are questionable. Also, it allows to answer questions, for example, is there evidence for multiple infections? Is there evidence that there are multiple infection sites, for example, upper respiratory tract, lower respiratory tract, things like that. It also allows to correlate sites which are changing with the sites that we predict as being under positive selection. So it provides a wealth of information, and I will begin by reminding you again that the state of SARS-CoV-2 data research is not great. So actually there is only small subset of data on which we can perform analysis I'm going to be talking about. So just to remind you, as of today, we have actually quite a bit an increase in the number of raw reads compared with the previous webinar, previous Thursday. So we have about 3,000. We still don't have raw data for most of the Oxford nanopore runs. There are about 17,000 complete assemblies in GZ8, so that's the discrepancy between the number of complete assemblies and the small number of raw data sets in the short read archives. And another thing which came to our attention last week, in particular the efforts of Sergei Kosakovsky-Fond, who's going to be giving a seminar in 2 weeks, is that the GZ8 data is not versioned. So sometimes there are changes to the sequences which are not reported. So one day you run analysis, you get one result, next day you run analysis, you get different results. So it's not something to be proud of, the state of SARS-CoV-2 data. But let's concentrate on the positive. So today we're going to talk about 4 things. I'm just going to briefly introduce how we call variants. Then Wolfgang will talk about how we actually do this in practice using Galaxy. Marius will talk about our automated procedure which runs the analysis Wolfgang will be describing every day upon release of new data. This happens automatically. And then I'm briefly going to talk about post-processing. So variant calling is conceptually is not a very difficult problem. You have reads, you align them to a reference, you find the differences. And there's a number of tools developed for this purpose. However, most of the tools that were developed for this purpose were developed in the framework of large international projects, which are necessarily human-based. For example, a project like 1,000 Genomes and its offshoots of this project. So the difference between human and SARS-CoV-2 is that SARS-CoV-2 is not diploid. So the distribution of allele frequencies is not discrete. It can have a given site, can have alleles with frequencies anywhere from 0 to 1. There is no expectation they can have homozygote or heterozygote of any kind here. In addition, because genome is relatively small, well, 30,000 base pairs, it's easy to get insane coverage. So some of the data sets have very high coverage. And when you have very high coverage, just by chance, you're going to get a substantial number of sequencing errors, which will look like variants. So at some point, for example, if you have coverage of, say, $1,000,000x, all positions will be variable. Because think about it, if we're looking at, for example, Illumina basis with quality of 30, which means probability of 1 error in 1,000. But if you have 1,000,000 reads, well, you're just going to have errors, which will look like variants. So with Illumina data, and everything I'm going to be talking today is based on Illumina, we don't yet have a good way to call variants from Oxford nanopore data because we don't have raw data. We don't have FAST5 data sets. So with Illumina, you can reliably call variants with frequency of as low as 1% maybe. So this is optimistic. In the framework of this discussion, we are going to set our lower thresholds at 5%. So we're being conservative here. And another issue is that SARS-CoV-2 resequencing is, it's an RNA-seq analysis problem because it's an RNA virus. So unless you're doing direct RNA sequencing with Oxford nanopore, you're going through cDNA stage. And depending how you do this, whether it's, for example, stranded or non-stranded RNA-seq, some of your downstream filters, such as, for example, considering strand bias of your variable sites, may need some adjustment. And so most tools are not geared up for this problem. And if we look at the latest literature from SARS-CoV-2 field, people use, for example, they use GATK to call variants on SARS-CoV-2. So they use all sorts of tools which are basically inappropriate for that particular problem. So what's the set of tools at our disposal? Well, there's a number of tools. And you can assess tools in two ways. Well, first, is the tool appropriate algorithmically? Does it do what we want it to do? And a second important consideration is so-called software rot. Software rot is a term, I think, coined by Sergei Kosakovsky. And that's basically when you look at the tool, before you decide whether you want to use it or not, look at GitHub repository. See, is it well maintained? Is it supported by a community and so on? So there are three tools which I'm going to be which I will be discussing today: Freebase, Mutect2, a part of the GATK package, and Freq. There are more tools, as I mentioned, but these satisfy this definition. They don't have software rot because they have quite a few commits. They're actively developed. For example, the last commit to Freebase was 8 days ago, for Mutect2 3 days ago, for Freq about 4 months ago, but it's still recent. There is a large number of commits. There are regular releases. There are more than one contributor. Many people fork this code. And so this is all sort of good signs. Freebase and Mutect are Bayesian variant callers, and they're haplotype-based variant callers. Freq is a frequentist. I'll talk a little bit about how this works, and it looks at individual columns in the alignment, so it ignores the neighborhood. So the difference between haplotype variant calling and column-based variant calling is that tools like Freebase, for example, they look at the neighborhood. And by looking at the neighborhood, you have advantage in figuring out which of the potential variants might not be real. Freq, on the other hand, doesn't do that. It considers every column independently, and it has a rather simple dynamic programming algorithm which allows you to estimate, which allows you essentially to calculate exact probability whether a particular variant, the reliability for a particular variant. So Freq is very simple. Freebase is more complex. Another reason why we're listing these two tools, because despite, I'm talking about Freebase and Mutect2, is that despite the fact that they were originally developed, again, in the framework of the 1,000 genomes for human, they have been modified to allow for calling variants in mixtures where the ploidy is not certain, when you have just, for example, a lot of viral particles floating. So Freebase has settings for dealing with this problem. And Mutect2 has recently gained a setting. It's called mitochondrial mode, which also allows it to do the same kind. So it makes it appropriate for viral mixtures and for. Sorry, I have construction going on here. Is it too bad? Can you still hear me?
Yeah, I think it's fine.
Okay. So, well, you know, the wonders of staying at home. So, and Freq was designed from the start for bacterial and viral variant calling. Okay, so how do I actually decide which tool to use? And in order to do that, so when you want to settle on a tool, you need to test it. So you need some data set which you consider truth. Well, in case of these kinds of samples we're dealing with, truth is actually very hard to come by because even if you're, say, sequencing a cloning vector, you know, PAC18 or PBR322, it still has some amount of variation. So these things are clonal, but they still vary. So in our case, what we've done is we took a data set which was produced from a duplex sequencing experiment. And a duplex sequencing is it's a molecular tagging approach which allows you to identify variants with very low frequencies. Again, in slides, this is an image from Schmidt et al. From Loeb's lab, and this method has been developed, and we're using it quite a bit. It's a good methodology when you're looking for variants which are below 1% frequency level, and with high enough coverage, you can get much lower than 1%. So what we've done is we have taken a data set from our recent publication, and this publication is a resequencing data for PBR322 plasmid. And there is a series of sites. So in this graph, you have 1, 2, 3, 4, 5, you have 8 sites. And these bars indicate counts of differences from the reference. So you can see that the different colors here mean different bases because these sites are naturally multi-allelic. It's not like you have G2A, you can have G2T or G2C, you have multiple variants. And that's their counts. These are not frequencies, this is absolute counts. And you can see that these counts are low. So these are variants at low frequencies. And we consider this a true set because we know these frequencies from duplex sequencing data. So next, what we've decided to do is to apply the 3 variant callers, freebase, mutect2, and lowfreq to this data to see which one of them will recapitulate that well. Please let me know if it's completely unbearable. I'll just run to a different room because I have noise canceling headphones. So for me, I don't hear it. Give me some feedback here.
Anton, just to let you know, your audio is really good. I don't see any background noise from your audio.
Okay, good. All right. So we applied the 3 variant caller to these data sets using a variety of settings. And it's an example. So if there's any graduate students listening to this webinar, don't put slides like this in your job talks because it's sort of very hard to read labels here. But well, it's not a job talk, so I guess I can deal with that. But what you need to notice here is that so these 6 panels correspond to 6 sites from this list from the previous graph, and in each panel, so these last 2 columns here, and here are lowfreq. So these are absolute counts of different bases from lowfreq. So these 4 are mutect with different settings, and these 2 are freebase with different settings. And you can see, well, that the lowfreq recapitulates that the best. Not sure. Maybe somebody needs to mute. Okay. So mutect2 actually behaves comparatively well. However, the problem with mutect2 is that it doesn't know how to deal with multi-allelic sites. So it simply computes the frequency based on the top allele. So if you have C and T, and C has a frequency, say, of 10%, and T has a frequency of 5%, it will report as though both of them have frequency of 10%. And you can see it here, for example. So in this case, the lowfreq differentiates between blue and orange, but for mutect, they're the same height. So based on that, and there will be more in this once we write this. You can ask questions, I can elaborate on that. So we decided that the lowfreq is the one to go. Of course, when you're evaluating, is my site really a good variant, you have to consider a lot of things. You have to consider various kinds of biases, such as strand bias, cycle bias, placement bias, allele imbalance, things like that. Again, if this is stranded RNA-seq data, then strand bias is not really going to help you because all your calls will be strand biased. But it's not the case for the data we've seen so far. And again, low-freq provides you, it scores sites based on some of these matrix. It also outputs some of these matrix, so you can for example, do additional filtering yourself. I should also mention that Freebase also outputs it. Freebase is probably the best tool in terms of outputting various kinds of matrix which you can post-process. But based on our experience in this framework, Freebase just calls too much. It calls the right sites too, but it calls it overcalls in our hands. Okay, so bottom line so far is that we are doing calling on individual data sets, resequencing from a particular individual, and we are using low-freq. And we came up with a workflow which Wolfgang is now going to show. And I need to stop sharing for that.
You can continue sharing it and just switch to the next slide, and I can just take over. So, as you already heard in the introduction, I'm working with Björn in the Galaxy-EU team. And part of my job there is to maintain our variant calling pipelines and variant calling tools in good shape. That means both in terms of technical quality, but also in terms of usability and user friendliness. So in this role, I'd like to shift gears a little bit, and hopefully, Anton has convinced everyone now that what we're doing here is good science in terms of the workflows and tools we're using. What I want to like to pick up right now from the first webinar is this idea shown here of bringing researchers to the data. And I think it was Frederick who presented the slide last time and who contrasted it with this traditional approach of bringing data to the researcher with the modern one of bringing the researcher to the data. And I guess he also made a strong point that this is good in terms of reducing redundancy and costs for everybody, both on the server side and on the end user side. And he also pointed out that the traditional approach doesn't really scale very well when it comes to large-scale data like this NGS sequencing data. Now, what I want to explore with you here is what this more kind of theoretical abstract concept really means in practice for you as researchers. And so when Anton and his team develop coronavirus variant calling workflows, what is it really like to use them through Galaxy? And what does the Galaxy framework let you do that might be very hard to do in the traditional way with just you and your work computer? So what's different? And for this, I figured out that the best way would be to just show Galaxy in action. And so what I want to do with you next is a short live demo on the Galaxy-EU server, which is kind of a risky thing, of course, to try in a webinar like this. So fingers crossed that it will work at this particular moment. It worked very well during testing, so I'm just hoping it will now. So I hope everyone can see my screen now. Okay, let's get myself a spotlight here. And then, okay, that doesn't work so well. Normal here. Okay, switch screens. Okay. So what you're seeing here is part of our EU Galaxy server data library, specifically the COVID-19 sequences subfolder and the Illumina data stored therein. So Björn and me have been working on offering this data through our public data library lately. And as a result, as a user of the server, you now have direct access to all such data that got deposited in the NCBI SRA short reads archive or the European Nucleotide Archive up until I think 2 days ago or so. So if you inspect this folder that you're currently seeing, you should be able to see that it contains like more than 3,000 data sets, which corresponds to roughly 1,600 or so individual experiments. And this is well mixed paired and single-end data. And the short demo I'm trying to give you is to just try and analyze all the single-end short-read data deposited and available for COVID-19 for SARS-CoV-2 so far live in this session. So let's see if it works out. One advantage of having this data here is that you can start analyzing it straight away without download worries. So the only thing you have to do is select your data of interest and let Galaxy know that that you want access to it through your current history. I'll show you in a minute what that means. So I prepared such a history already. So it's the one that's shown here. And it currently holds only one data set that I already uploaded from our data library. That's the GenBank reference file of SARS-CoV-2, so that we get a hand on the reference sequence and an annotation of it. And now back to the data library. Now I'm just going to select all the Illumina data in that library. And I go to export to history as a collection. So a collection is kind of a container for many data sets in Galaxy, and you'll see it soon live. Then it asks me what collection type I'm trying to build, and I say I want to specify my own custom rules, because what I want to do is I want to filter out the paired end data and just work with the single end data in that folder. So I go to from rules, and then I have prepared this demo history here for this purpose. So that's where the data will end up in. I just go to continue, and now I'm seeing all these data sets, and now I'm applying rules. And for the sake of time here, I already copy-pasted rules in JSON format that I just reuse here. This is actually also a good thing to do because it ensures reproducibility even for this data import step. So by reusing that same rule, you can get a hold on the same data again. I apply that. And when I do that, Galaxy splits my data into several columns, and it now only retains the single-end sequencing data, and it will extract the SRR identifiers from the data set names, and these will become the element names in the collection. So now I still have to provide a name for this collection. Let's go with Illumina single-end data for this purpose. And then I can create that collection. So what happens now is that Galaxy in the background will give me access to all this data through my pre-prepared history, which actually does not mean that there's any data duplication going on, but it's basically just bookkeeping in the Galaxy database to take note of what's accessible through my history and whatnot. This will take a couple of seconds. We can actually switch over to my history and just try to see the progress there. So there's data coming in already. And there should be a collection item very soon. Maybe it ended up at the bottom. No, it didn't. Okay. So let's try refreshing again. Well, I don't know if this worked like expected, but let's see. So I have 236 data sets in that history right now. So now I'm trying to analyze that data with one of the workflows that Anton and his team built. So I'm going to workflows where I have imported the single-end data variation analysis workflow already. And I simply try to run it now on this history that I've just populated with data sets. do that, after a moment of thinking, Galaxy should offer me to run this workflow. And it can't find a dataset collection, which is kind of bad. So basically all these, I know why, because I'm in the wrong history. That's a bug that happens occasionally. I'm sorry for that. So let's just switch to the right history. Truly sorry. So now we have that Illumina dataset collection that I was building with 236 items in it. That's the right thing. And now if I run that workflow again, now I have the chance to input as my dataset collection that Illumina single-end data with the 236 single-end public data samples and my reference genome for mapping. And now I can simply run that workflow. So, and what just happened is that I told Galaxy to run this workflow of 12 individual analysis steps in parallel on all 236 samples in that collection. So this is certainly something you would be hard pressed to achieve on your own machine locally. And this is all the single-end sequenced data that is currently available in public places for SARS-CoV-2. So what we'll get out of that, hopefully, is a list of variants of all these different samples. And of course, this will not finish instantaneously, but Galaxy now has to dispatch all these jobs on our production server, find slots for them on different machines in the cluster, and read and collect the data again. So it will run for a while, but we can hopefully have a look at the end of the seminar what happened to it. So with that, I'm at the end of my live presentation, and if you can just switch back Anton into presenter mode, then I can show you in more detail what just happened. So
thank you very much.
Where was it? Slide. Maybe go to the workflow slide that I put there. This one, yeah. So thanks. So this is the workflow I just started. So here's how you could run it from the workflow editor. You could also run it like this by pressing this button, the play button. So it's, as I said, a multi-step workflow, which first does quality control and read trimming through the FastP tool. Then it maps the single-end reads using Bowtie as the read mapper. It marks and removes duplicate reads to make variant calling and frequencies of called variants more reliable. It does realignment to improve the quality of the variant calling. It then calls the variants, and this is where LowFreq really comes into play. This is LowFreq's core business, calling the variants. And then it annotates all the variants it found using snpf and that genbank file that we put in it as the second input, which has the annotations, the known features of the SARS-CoV-2 genome, and snpf is using them for annotation of variants. That's the rough overview over this workflow, and this very workflow is now currently executing on the EU server 236 times in parallel to analyze all that data. So this workflow is not the only one, of course, that the COVID Galaxy project developed, but there's many more. For example, there's a very similar workflow for variation analysis on pad-end data. So instead of the 236 single-end data sets, we could have analyzed the 1,200 or something pad-end data sets as well. There is also an assembly workflow for which we even have a nice tutorial on the Galaxy training pages. So if somebody wants to really get into Galaxy at a slower pace, first experimenting a bit with it, this might be a good starting point to follow that training. And yeah, that's some of the workflows we offer. So when this workflow that I just launched finishes, that's in the next slide, it will give us a very tidy view on what it did in the history. So for each step in the workflow, we will generate another collection of 236 data sets, which represents the analysis step conducted for these 236 samples. So there will be 236 SNP-FF data sets, for example, in the SNP-FF collection with the called variant effects. Yeah, so that's really easy way to parallelize your work and to work with a large number of samples. What's equally important though is that Galaxy offers everything for sharing your analysis, for documenting it, for making it reproducible. So you can share your analysis or your workflow with individual users or also publicly on public servers. You can even download workflows to share them across public or private Galaxy instances. And what I just showed you should work on ever-growing number nowadays of public Galaxy servers, including the US server, the Australian server, the Belgian public server, and French public Galaxy instance, and use Galaxy-EU, of course. You can reproduce every single step in this workflow because all parameters used to run each job are automatically documented for you, and you can use these recordings basically to rerun every previous job with identical settings again. Yeah, and as I said, we will inspect this more at the end of this webinar and see what EU produced for us until then. And that's as far as you can get as a regular end user with Galaxy. So what Marius is now going to present you is what you can do on top of that if you actually are in control of your own Galaxy instance. So Marius, are you ready?
Marius, you have to unmute your microphone.
Okay. Can you hear me?
Yes, now, yes.
Okay. Thank you, Wolfgang. Yeah, that's a great transition, except that what I'm going to talk about is applicable to all Galaxy servers. But let's start. So we have now this nice workflow, and we started using it from the beginning of February until now, and we've regularly updated the results, redone the analysis. And in the beginning, that was all fine because we were only dealing with irregular updates of a couple of samples. But as you can see here on the right, the volume of data deposited and the depositions both increase in frequency and amount. And as Anton has said, a lot of the, ideally all data would be deposited in one of the raw sequencing archives. So wherever you are, there is one that is close to you. So there is the sequencing read archive run by NCBI. There is the European nucleotide archive that's run by I always forget which one it is, ENBI. And there is the Japanese DNA sequence read archive. So they all mirror and distribute data. And what is great about them is they have been in operation for many years, and they are very scalable. They can handle the largest amount of data submissions. And again, they've been around forever, at least being a relatively young researcher since I started, they've been there. And yeah, this is really important. That is public infrastructure that we can use, and that's a bit different from GIS8. So anyone can get the data. Next slide. Okay, so one of the goals we had was Something's wrong with the slide, I think. Ah, there it is. Yeah, so we had a couple of goals to make all of this available as quickly as possible. So ideally, whenever there would be new deposition of data, we would find the new data, so find the new accessions for all the new published SARS-CoV-2 data sets. And then we would run a workflow on the new accessions. We would want this to be tolerant to failure. And very importantly, we want this to be transparent and public. You should be able to see what we have done, when we have done, how we have done it. And then ideally, provide this infrastructure for everyone to use and build on. Next slide. Okay, so the first part of this is we need to find new accessions. And as I said, all these sequencing archives, they provide stable APIs and they exchange data one with the other. So data deposited at the ENA, you can find at the SRA. Data deposited at the DRB can also be found this way. So that's great, and you only need to query one. So there is a nice Python library that wraps the API that the sequencing read archive provides. So this one's called PySRADB. And it has a nice command that is PySRADB search. And then you can search for a taxonomic ID. So in that case, we would just use the one for SARS-CoV-2. And to automate this, we've put this on a GitHub workflow. So these are workflows that run directly on GitHub and have access to your data in your repository. And they can also submit data to your repository. So on the left, you see a run. So we run this every night. And we look for new data, and new data in our case meaning new assembled genomes and raw read data. So this is a run where we found both new genomes that you can see in the upper half and new accessions that you can see in the lower half. And when there is new data, it creates a pull request on the SARS-CoV-2 GitHub repository. So this bot opened this pull request that you can see on the right. So that's the first step. So next slide, please. And then we want to analyze that, right? So we have this great workflow that Anton and Wolfgang will describe. And we've optimized this a little bit for higher volume. So it takes as input the accessions and the GenBank file from which we can, these are the two starting inputs to the workflow. That's all we need to get to the result that Wolfgang described. So then it goes into a cool feature that Galaxy has, which is including other workflows. So we have one workflow that just downloads the accessions from the SRA. This one is quite interesting because it's fault tolerant. There is the SRA toolkit that works fine most of the time, but when you start downloading thousands of accessions, one or two of them will fail. And so this workflow does them all in parallel, so you only download one accession, and then at the end, the failing ones it reports, but it will continue the analysis. From there, we can split the data sets into single end and paired end because they're treated slightly differently. So you see the parallel accession download Illumina. The outputs go either into the COVID-19 PE variation for paired end, or you go into the single end variation workflow. At the end, though, we can interpret them in a relatively similar manner. So these workflows, the output of those two variant workflows, we can combine, and ultimately we end up with a single table and multiple reports that we can investigate. So next slide, please. Okay, so what is important is that these workflows are fully annotated. So workflows, Galaxy workflows, can be thought of as a program or as a tool. So that means you need to describe all your inputs and all your outputs. And if you do that, good things happen. So here's an example of how it looks like when you run the workflow, or depending on which Galaxy server you use, it might look a little bit different. So I already mentioned there's these two inputs that you can see on the left. So these two files have to be selected. And that's basically it. After that, you hit run workflow. But we want to do this automatically. We want to do this when new data becomes available. So I've written a small tool called workflow to executable that you can see on the right. So this is the basic workflow. So you can type --help, and it will tell you what you have to do. So basically, you run this tool, you give it a workflow ID that you can find in the Galaxy interface, and you give it the Galaxy instance where that workflow is hosted. And we'll take that workflow and write an executable script. That's workflow to executable. So you see that in the second command that I ran. So it's workflow to executable, the ID of the workflow that uniquely identifies the workflow and the Galaxy instance. And then there's an additional switch and that workflow. So the entire workflow will be actually in that script. And then -s means save the script to that path. So save it to cov2 variant workflow.py. And this Python file you can run as any other Python program. And it also describes itself because there are fully described inputs. It's very clear what is needed. So if you type Python cov2 variant workflow.py --help, it tells you the little description about what this workflow does, which is from download single and parent COVID variation workflow. And then you see the options. So it requires a -- genbank file, and it requires an accession file. So these are the accessions that are needed. So I've shown you how to use it. So the last example is how you actually run this. So this looks like a normal command line tool. And that's great because we can use that now in the infrastructure, in the GitHub application, in the GitHub workflow that we built. Next slide, please. So here's an example. So together with finding the new data, we now also trigger this workflow. And so we're still developing these things. As I said, we slowly started scaling things up. So this is a relatively new thing. But here you see that 5 days ago, we triggered this workflow. And then it ran this workflow on usegalaxy.org. It created a history with about 11,000 datasets that constitute the entire analysis. And at the end, you get a single table out. Of course, there's also a multi-QC report with all the different things, quality control things that you want to look at. There's also VCFs and BAMs for genome browser visualization. But I think the most important output here is the summary table of all the variants found. And this history will be public the moment the workflow is started. The history will be immediately public. You can follow it. You can go on GitHub. You see, okay, here's the new workflow coming in. It will print out where the history will be published. And you can go there. You can see the progress. And most importantly, since it's a shared history, you can import it and you can build it. And you can do your own analysis with this. If you have a great idea of what to do next with this data, if you have a certain question that you want to ask, you can use this. Next slide. And Björn asked me 2 minutes before the webinar started if we can also do this in usegalaxy.eu. And of course we can. So this was also triggered by the same script. Next slide. So here I want to make a few points. So this is unfortunately not the last version apparently. But what I want to say is that this infrastructure is free and it's public. You should all go use it. It can be run against all modern Galaxies with a small bracket that the tools need to be installed, but this is the case for the big public instances. So usegalaxy.eu is galaxy.org, usegalaxy.org.au. There are no special permissions required. So in order to run this entire workflow, we needed to have a little bit more storage, but otherwise anyone could run this. You can import the history and run your own analysis. So I think this is really important. It highlights that you can collaborate efficiently by using Galaxy. It builds on many years of expertise in development. And as a public web server, Galaxy is ideally suited for collaboration. So with that, I will hand back to Anton.
Thank you. So we have 5 minutes. I will just quickly mention, well, I'm going to switch to. OK, so as Marius said, whatever you run, either through a graphical interface or if you want to script it through Galaxy API, but in the end, you're getting a list of variants. But when you're trying to write a paper or present a talk, you're not showing tables of variants. You're going to summarize them somehow. And that summarization is ad hoc. Some people want to see this. Some people want to see that. And in order to accommodate sort of these scenarios, what we've done is so Galaxy integrates so-called interactive tools. An example of interactive tools are, for example, Jupyter or RStudio. There are other examples. So what you can do directly in Galaxy is that what I have here, I have a Jupyter instance inside Galaxy interface. And inside that Jupyter instance, I can pull in data sets from my Galaxy history and perform essentially any analysis you want to do. In this case, I'm using Python. And one of the analysis I've done is, this is, for example, a distribution of two kinds of substitutions, two kinds of variants, non-synonymous substitutions that change amino acids and nonsense substitutions, which create stop codons. And of course, you can see that low frequency cloud of orange of these nonsense substitutions, most of which are probably spurious and spurious artifacts. But you can also see these sort of columns of where a single site is changing repeatedly in multiple samples. And for example, in case of this substitution inside of ORF1b, you have changes which vary anywhere from about 60% in frequency all the way up to fixation. So you have a number of these sites. And this is something we're going to have in subsequent seminars where we combine this information with information about sites under positive selection and try to make predictions of where the virus is going and what these sites can potentially do. But the important thing, what this webinar was about, one, you can use Galaxy to run analysis of this sorts yourself, but you can also use the data we have produced. And so if you actually, well, that's the wrong stuff. So you can always go to our GitHub repo. It's Galaxy Project SARS-CoV-2. And so let's go here, for example, and there is a genomic section, variant analysis. And here you will find all the VCFs. So these are indexed VCFs for all the samples we analyzed so far. And again, this is updated essentially daily. Plus, you have the variant lists, both filtered, for example, in this case, and at 5% frequency. These are very large files, and also unfiltered, where you have all variants. We can also make accessible BEM files and so on. So this resource is for both if you want to analyze your data, new data, or if you want to make synthesis analysis of existing already analyzed. You can do both things. I'm just gonna finish here.